Hello, and welcome to our webinar, Pushing the Boundaries of Gene Sensitivity with the Chromium Single-Cell Gene Expression Solution V3. I'm Shauna, the 10X Community Manager and your moderator for today. Before we get started, let's go over some quick housekeeping items. Our speakers, who I'll introduce in a minute, will be giving a presentation followed by an open Q&A session. We're recording this session and we'll make the recording available on the 10X website shortly. Please note that all attendees are on mute, and if you would like to ask a question, please select the Q&A option on the bottom of your screen. Selecting the Q&A option will open the box where you can type your question. And finally, if you're interested in tweeting during the session, please use the hashtag #Celebrate. Now, let's meet your speakers for today. Up first is Jens Dorothy, a product manager here at 10X. Jens holds a BS in Medical Biotechnology from Technical University in Berlin and a PhD in Biomedical Engineering from Stanford University. Focusing on stem cell research and RNA biology, Jens worked with various single-cell technologies on and off the bench for 6 years before joining 10X Genomics. Jens is now the product manager responsible for the single-cell gene expression solution. Joining Jens will be Jessica Terry. Jess received her undergraduate degree in forensic science and her PhD in analytical chemistry from Deakin University in Australia. Since joining 10X over 5 years ago, she has worked on the GemCode and single-cell gene expression solution. And now I'm going to hand it over to Jens so he can get started with the presentation.
Thank you, Shauna. Hello, everyone, and thank you for joining us today as we give you an update on our new version 3 of our single-cell gene expression solution. We're really excited to review with you the new solution, its workflow, and some of the new data that highlight all of our product improvements. For those of you who have listened in on our other webinars during the summer, welcome back. Biology, and especially mammalian and human biology, is incredibly complex. There are thousands of differentially expressed genes to millions of T and B cell receptor pairs and billions of DNA bases in the human genome that are present in billions of individual cells that define each and every one of us. Key in understanding these complexities is to profile individual genes, the genome, and the full immune repertoire at the single cell level to drive discovery research and ultimately accelerate disease and therapeutic studies. At 10X, we strive to build solutions and develop tools that enable you to uncover the full complexity of human biology. 10X Genomics has a comprehensive list of solutions that enable cellular phenotyping at high resolution. This includes our linked-read genome sequencing solution, our single-cell gene expression and immune profiling solutions, and our single-cell CNV solution. We've recently expanded our product portfolio with the addition of our single-cell attack solution and have introduced our feature barcoding technology, which enables simultaneous assessment of gene expression and the immune repertoire along with additional features. I encourage you to listen into our recorded webinars by visiting our 10X Genomics website to learn more about each of those new solutions. Today, we'll be talking and focusing on our single-cell gene expression version 3. Our single-cell gene expression solution enables the profiling of thousands of individual cells based on their transcriptional profile. This is in contrast to traditional bulk sequencing techniques where the complexity and heterogeneity of a given sample is masked by an average of your gene expression across your sample. And so as a consequence, the only readout we get from microarray or other bulk RNA-seq studies is a single sample readout that represents the combination of many gene averages. With the scale and ease of use that 10X has enabled, you can profile tens of thousands of cells and multiple samples at a time to reveal the full cellular heterogeneity on a transcriptional level. This unbiased approach to profile thousands of genes across thousands of cells enables you to resolve subclusters and identify rare populations within a complex sample such as cancer or other tissue types. To the right is an example of PBMCs, peripheral blood mononuclear cells, that are profiled on a single cell level. What you're looking at is a two-dimensional view of a T-SNE plot where each dot represents a cell that is clustered based on the transcriptional profile. Two dots far apart from each other have very distinct and different transcriptional profiles. Each cell is also color-coded based on specific marker genes, which, as you can see, allows you to resolve distinct cell subpopulations and transient cell states that are impossible to detect with conventional gene expression assays. Our current single-cell gene expression solution has enabled researchers to do discovery research at scale and within many different application areas. Unbiased single-cell RNA sequencing, for instance, has led to the identification of novel, previously unknown, and very rare cell types in well-studied biological systems that play important roles in disease biology, like cystic fibrosis. Single-cell RNA sequencing is also a great tool to resolve the full heterogeneity of complex samples, such as the brain, and to discover transient cell states and distinct cell types. Last but not least, the Chromium single-cell gene expression solution has helped accelerate cancer research by unraveling the cellular identity and resolving the tumor microenvironment of different cancers, including renal cancer. The core of the 10x Chromium system and solution is the microfluidic partitioning of reagents and cells into single nanoliter droplets called GEMs, which stands for Gel Beads and Emulsion. This is enabled through the Chromium controller and a microfluidic chip. What you see in this video is how each of these components are brought together. The gel beads flow from the left to the right and are mixed with the sample and reagents before they are surrounded by partitioning oil to form the GEMs. The key to our system is its speed, the barcoding of each reaction, and the efficiency of the GEMs being made to only contain one cell for some of the highest throughput, cell recovery, and lowest multiplet rates in the industry. And so our new single-cell gene expression solution provides an unbiased approach to assess the single-cell transcriptome, which ultimately leads to novel, unexpected biological discoveries. It's a solution that is compatible with whole cells and nuclei and really enables the interrogation of a broad range of cell types and species, including cell lines, primary cells, and dissociated tissue. Features of the solution have not changed compared to our current V2 chemistry. It's a ready-to-use solution that starts with specific guidelines on how to prepare your sample of interest and ends with software tools that allow you to conveniently analyze and visualize your single-cell data. The single-cell gene expression solution is strong in cell and sample throughput, has no major limitations in cell size, and has industry-leading cell capture rates of up to 65%, which is an important feature for those working with limited numbers of cells. There's a number of improvements that come with the new single-cell gene expression solution version 3. The new version 3 chemistry is fully optimized, resulting in a significant increase in performance both on the assay and software side. We've optimized our reagents and workflow to achieve an up to twofold increase in gene detection, potentially decreasing sequencing requirements. The new version 3 single-cell gene expression solution also comes with the added feature barcoding technology capability. This allows for the simultaneous assessment of gene expression, protein abundance, and CRISPR-mediated perturbations, among others, from the same cell. We will not cover any details on this added capability in this webinar, but I encourage you to listen into our recorded webinars or watch our How It Works videos at the 10x Genomics website to learn more about the feature barcoding technology. There's also a number of software improvements, including an upgrade of our Cell Ranger software pipeline, which has improved in detecting low RNA content cells. Jess will outline this in more detail later. In this set of data, both of our version 2 and 3 gene expression solutions were run with a complex sample type, in this case, mouse embryonic cardiomyocytes. You can see in the chart that the gene sensitivity is twofold greater at the standard sequencing read, which is 50,000 raw reads per cell. This improvement in gene sensitivity allows us to detect more genes per cell without the need for additional sequencing reads. The new version 3 is more than just about increased sensitivity, though. It also provides you with an increased flexibility of your experimental design. Compared to version 2, one could increase the number of cells within the same overall experimental costs. Alternatively, you could target the same number of cells and achieve the same gene detection metrics from today with more than half of your sequencing costs reduced. Lastly, you could push the boundaries of this new solution and sequence even more to maximize the number of genes detected. Many dials are now available to you with this improvement in performance for you to run your experiment the way you need to. I'll hand it over to Jess now, who will walk us through the workflow of the solution before reviewing some of the data that she has generated in-house.
Thank you, Jens. So this is an overview of our single-cell gene expression workflow. Hopefully this looks very familiar to many of you, as this is the same workflow that we used for our single-cell 3' V2 product. Single-cell 3' V3 gel beads are master mix containing cells and oil are loaded onto a microfluidic device and partitioned to create gems, or gel beads in emulsion. Immediately following gem generation, the gel bead is dissolved, primers are released, and any co-partitioned cell is lysed. Incubation of these gems produces barcoded full-length cDNA from polyadenylated mRNA. After incubation, gems are broken, and the first strand cDNA is amplified via PCR. Library construction is then performed, creating a standard illumina paired-end construct ready for sequencing and data analysis. Our updated reagent kits allow greater flexibility for sample processing. They contain, as you can see here, we have a gem kit that contains all of our components for gem generation and cDNA amplification, a library kit that contains all of the reagents needed for library construction and is available for purchase separately if required, our single-cell 3' V3 gel beads, and a feature barcode library kit, which is optional for purchase if you are interested in performing feature barcode technology. I'd like to take a moment to highlight our single-cell 3' V3 gel beads. These gel beads enable feature barcode technology and also our standard gene expression workflows. Each gel bead contains barcoded oligonucleotides that consist of a TrueSeq read 1 sequence, shown here in black, our 10x barcode, which is a 16 base pair barcode now drawn from a pool of 3.6 million defined sequences that are used to index the cell, our UMI, shown in orange, which is now 12 base pairs in length, and a 30 base pair anchored poly-DT tail that is used to prime polyadenylated mRNA transcripts. In addition to this poly-DT primer, we now also have 2 additional primer sequences that enable capture and priming of feature barcode technology compatible targets, shown here, capture sequence 1 and capture sequence 2. These primers are not used in the single-cell gene expression only assay, which is why they are shown or why they're kind of blurred out here. Our single-cell 3' gene expression libraries are compatible with Illumina sequences. They comprise standard Illumina paired-end constructs, which begin and end with a P5 and P7. The 16 base pair 10x barcode and 12 base pair UMI are encoded in read 1, while read 2 is used to sequence the cDNA fragment. Our sample index sequences are incorporated as the I7 index read. You'll note here that our read 1 length has increased from 26 base pairs with our V2 chemistry to 28 base pairs with our V3 chemistry, and this is due to the slightly longer UMI. So now I'd like to talk to you about a couple of data sets that we generated while developing this product. During this process, we validated many samples internally. We use cell lines, both suspension and adherent, derived from both human and mice. We work with primary immune cells, particularly peripheral blood mononuclear cells or bone marrow mononuclear cells, again derived from humans or mice, and also dissociated primary tissues, particularly embryonic mouse brain tissue, embryonic mouse whole heart tissue, mucosa-associated lymphoid tissue, which is a form of extranodal B-cell lymphoma, and glioblastoma multiforme. To assess the technical performance of our system, we prepared a mixture of human HEC293 T cells and mouse NIH3T3 cells and performed a cell loading titration experiment across 3 different cell loads. As you can see from the plots below, there is clear separation of the human cells along the x-axis and the mouse cells along the y-axis. You can also see the multiplets shown here in grey that form between these 2 axes. We have a low doublet rate that is equivalent to 0.8% per 1,000 cells at each point in the loading titration. Next, I'd like to talk to you about profiling complex primary cells with our gene expression solution. We typically work with PBMCs because they are a complex mixture of different cell types. They're well studied and readily available primary cells. For this particular experiment, we purchased fresh peripheral blood mononuclear cells from a healthy donor from all cells. These cells were prepared following our demonstrated protocol for fresh frozen human peripheral blood mononuclear cells available on our support website. 2 cell loads were targeted, and in this particular case, the same tube of cells was used to prepare single-cell 3' V2 libraries alongside single-cell 3' V3 libraries. As you can see here, all major blood cell types are discerned using both of our chemistries. You can see we have very similar proportions of each of these cell types between both of the chemistries. We can see our monocytes shown here in green, our dendritic cells shown here in purple, our B cells in yellow, our large T cell cluster in pink, and our NK cells in blue. When we break out the plots and look at these key markers by themselves, you can see increased sensitivity for each of these key markers, particularly when you're looking at with our V3 chemistry. You can see more intense UMI counts coming from our T cells, our B cell population, our monocyte population, our natural killer cell population, and our dendritic cells. When you compare these samples head to head at a normalized read depth of 20,000 raw reads per cell, we can see an increase in the number of median genes detected, going from almost 1,800 median genes to just over 2,300 median genes. You will also notice a significant decrease in sequencing saturation from 54% to 39%, which is fairly remarkable for this cell type. When we profile our 1,000 cell sample and we sequence out much deeper, you can see that we're gaining significant increases in the number of median genes. We start with just over 1,000 median genes for single cell 3' V2, and at our standard sequencing depth of 50,000 raw reads, we see an 88% increase in the number of median genes detected, with almost 2,000 median genes detected per cell. This is also accompanied by this drop in sequencing saturation from 87% to 67%. Similar increases are also observed for our UMI counts. As you can see here, we're obtaining almost 3,500 median UMIs per cell at 50,000 raw reads with our single cell 3' V2 product, and that has now increased to over 6,600 median UMIs at the same fixed read depth with single cell 3' V3. Next, I'd like to talk to you about profiling complex primary tissues with our gene expression solution. As you can see in the panel on the right, we have a TSNE plot representing 1.3 million cells, which we prepared in 2016 when we launched our single cell 3' V2 product. While we haven't performed an experiment on that scale for single cell 3' V3, we've worked with the same type of tissue that was purchased from BrainBit and prepared according to our demonstrated protocol for mouse embryonic neural tissue. Again, 2 cell loads were targeted, and the same tube of cells was split between our V2 and our V3 chemistries. You can see that by looking at our gene expression markers, we're highlighting both neuronal and glial clusters. While the shifts in intensity may not be as obvious as with our primary PBMC samples, we are seeing gains in the number of median genes detected per cell and drops in sequencing saturation. When you compare our chemistries looking at the 1,000 cell samples, again, you're seeing significant increases in library complexity. We're seeing an increase in the number of median genes detected from just over 2,000 with our V2 product to just over 3,100 median genes per cell. And we're seeing slightly larger gains with respect to our number of median UMIs, with increases from just under 5,000 median UMIs per cell to 8,744 median UMIs at the same fixed read depth of 50,000 raw reads per cell. The last data set I'd like to talk to you about is profiling dissociated tumor cells with our gene expression solution. As you can see in the panel on the right, a tumor sample such as glioblastoma is a very heterogeneous, complex sample that contains many different cell types. Glioblastoma is the most aggressive primary brain tumor in adults, and it's a fast-growing glioma that develops from glial cells, in this case astrocytes and oligodendrocytes, that support the health of nerve cells within the brain. The tumor microenvironment contains many different non-cancerous cell types in addition to cancer cells, including endothelial cells, parasites, fibroblasts, and immune cells. We obtained the sample from Discovery Life Sciences. It came pre-dissociated. The cells were thawed and prepared following our protocol for human peripheral blood mononuclear cells. As with our other experiments, we targeted 2 cell loads, 1,000 cells and 10,000 cells, and again, the cell sample was split between our 2 versions of the chemistry. As you can see, even just looking at the default graph-based clustering resulting from analyzing the data with Cell Ranger, there is a high level of substructure present within the sample and very similar proportions within our single cell 3' V2 and V3 chemistries. You can see increased sensitivity for key markers within the tumor microenvironment when you look at several select markers, particularly our T cells, our infiltrating tumor cells, you can see in our middle panel here, our oligodendrocytes, and our vascular cells. Again, when these chemistries are compared at a fixed read depth of 20,000 raw reads per cell, you can see an increase in the number of median genes detected and an increase in the number of median UMIs. When the 1,000 cell samples are sequenced, you can see that while we do get gains in sensitivity, they aren't as pronounced as our very small primary cells, our PBMCs. Now, one of the reasons for this is because we're sequencing, the sample is so complex with a combination of both high and low RNA containing cells. Our sequencing saturation at 50,000 raw reads per cell is only 41%. But despite this, we're seeing an additional 400 median genes per cell and over 1,000 additional median UMIs per cell. As Jens said earlier, we've made improvements to our cell calling algorithm, and samples like the glioblastoma are a perfect use case example for why improvements have been made. In this particular example, we simulated a very heterogeneous sample by combining HEC293 T cells, which are large cells containing about 15 picograms of RNA per cell, with PBMCs, very, very small cells containing about a picogram of RNA per cell. As you can see in the plot on the left, Cell Ranger with Cell Ranger 2.2, only the large mode cells, in this case the HEC293Ts, were called, and the small mode cells, the PBMCs, were not called as cells. In many cases, a customer would have used forced cells or the forced call option to be able to drag the cell call down to where they needed to be. With Cell Ranger 3.0, this no longer needs to happen, as the algorithm has been finely tuned to now be able to detect differences between the large mode cells, the small mode cells, and the background. And as you can see, both of the cells in this sample were now picked up using Cell Ranger 3.0. The final thing I'd like to mention is that if you really want to push the boundaries of our single cell 3' V3 assay, you can sequence your samples out to extremely high read depth. As you can see here, we've sequenced out our barnyard samples out to almost 1 million reads per cell, and we're obtaining almost 10,000 median genes with our single cell 3' V3 solution and just over 7,000 median genes for our NIH3T3 mouse cells, which is extremely impressive. And with that, I'd like to open up for our Q&A session, and I'll hand you back to Shauna. Thank you for listening.
Thank you, Jens and Jeff, for a great presentation. Before we get